There we go. Looks like people are starting to join. Hopefully you all didn't have to run off and microwave leftovers like I did. Um, all right, so I think we can go ahead and get started here. We may have some more people joining as we go, but um, thank you everybody for rejoining for our guest presentation today. This will be module 5.2.3. We have our guest speaker, Bharat Vivan, from Vanderbilt University. He's going to be presenting today on a model he published, it was last year, correct? On the senescent mesothelial matrix, which accentuates the colonization by ovarian cancer cells. Um, so Bharat is a previous CompuCell workshop attendee. He attended in 2022 and had an excellent hackathon experience, so excellent apparently that he decided to continue using CompuCell, which is what we always like to see. So he is coming back today to show you his work, which, like I said, was published last year, and I think is an extension of what he started to do in the hackathon. So he will walk you through kind of his experience with that, as well as the model itself today. Excuse me, sorry. So just as a reminder, this session is currently not being live streamed, but it is being recorded and it will be published to YouTube over the weekend, this weekend, so that people can view it who weren't able to attend. So do just keep in mind that the session is being recorded, although we are not currently being live streamed. And as always, we like to thank our funding providers through the NSF and previously through the NIH, as well as our support networks through the IUB Biocomplexity Institute, as well as the OpenVT and Glimprint initiatives. So I will go ahead and turn things over to you, Bharat, and you can get started when you're ready.
Awesome. Thanks, Hayden. Yep, I'm ready to start. I'll just share my screen. Okay, I hope everyone can see the screen. Just pull out the laser pointer. Oh, the the red won't work because I have a lot of red stuff.
We can see your cursor. It's
Oh,
visible now.
you can? Okay, awesome.
Yeah.
Awesome. Uh, Never mind. Okay, we'll get started. Today I'm going to be talking about some of the research that I did in my bachelor's and my master's under Professor Ramred in the Institute of Science. And then I learned CompuCell 3D while I was an undergrad and had a great experience in the hackathon with some of my friends and obviously the support people that I had from CompuCell 3D to help me navigate things, learn new tricks, a whole new platform. And then I implemented that in my research, and it turned out great. So yeah, I'm just going to talk about mostly the modeling part, but we will need a little bit of experimental background. And I think I really, after this experience, I really like the idea of integrating modeling into my research, although I'm a hardcore experimental biologist. Awesome. So this is just a video which should be playing before, but I forgot about it. On the left is the experimental setup that I was trying to mimic with the computational modeling. And on the right, hopefully, in some time, I'll get clear to you what cells are moving around and what is going on. Before I get started, I just want to acknowledge the support and help of everyone in Ram Rebhard's lab, who has been really nice to me. As a PhD, I don't like undergrads working around, but they were very nice to me, so I really appreciate it. I see something popping in the chat. Oh, awesome. Thanks, Hayden. Feel free to actually unmute yourself also and just scream out a question if you have. Um, I don't mind that. Cool, thank you. Um, so thank you, Ramres Lab, for helping me out throughout my undergrad journey. And then my awesome hackathon teammates, Lakshay and Uday, who helped me out. They were hardcore coders, so they helped me a lot with the coding stuff. And then I learned CompuCell 3D a little bit from them, a little bit from the CompuCell team. And then I also want to thank the CompuCell team, James, who actually took time to um, help us out with our queries and questions during the hackathon and during the lectures. Hayden for organizing all of that and being a support friend all the time. And Pedro, who is also like a teaching assistant, and he helped me a lot during the hackathon period. And everyone else, all the course instructors who made it a great experience, and the guest lecturers who really taught me what are the innovative ways you can actually think of using modeling into your experiments, as well as the other TAs and the students, my summer school mates who made it very interesting and shared each other's science, and it was pretty fun. Awesome. Let's get started. So this is going to be one of the lectures which is on the longer side that I've given before, so I thought it'll be useful to have an outline. We're going to start with project background, and then a little bit of what were the experimental findings, how did I come up with the project, how did it go, how did it move forward, what were the challenges, and then we're going to talk about how I implemented the modeling at that stage and how did I figure out that this could actually be a good modeling problem and how one can actually think of modeling problems and what are the kind of basic things which are needed for modeling. And then we'll move forward to choosing a modeling problem and what are the things which are required. And then I'll go ahead and actually introduce you to the model. We'll look into the code. We'll look into how the model works. We'll look at what are the outputs of the model, how the model is implemented. And then to make it more fun and hands-on, we're going to try out a lot of exercises. Some of them optional, some of them which I would like you guys to work on and actually collect data. All right, so a little bit of background. As you know from my talk, I'm going to be working in the regime of I'm going to be talking in the regime of ovarian cancer and aging. So first of all, how does ovarian cancer really originate? So these are the ovaries right here. And I'm going to be specifically talking about a type of ovarian cancer called the epithelial ovarian cancer, which is one of the most aggressive forms of ovarian cancer, where the outside of the ovary actually has these epithelial lining and the cells due to some stimuli, maybe chemical mutation or any sort of mutation, gets kind of transformed and they get exfoliated from the ovary. And now once they have actually exfoliated and detached from the ovary, they're free to move literally any of these tissues. One of the common sites where ovarian cancer ends up getting stuck and metastasizes to form secondary tumor sites is this peritoneal cavity, peritoneum, which is shown here, pelvic peritoneum, and the omentum, which is like a fat tissue which guards your intestine right above it. So these are the two major sites where ovarian cancer, once originates, likes to metastasize. Now, one of the things which we observed, which comes from an epidemiological study, which is shown here actually, is that when you look at metastasis as well as origin of ovarian cancer, you see this very extreme trend of cancer, first of all, occurring and originating in elderly women. When I say elderly, I mean, say, beyond 55 or something. And not just that, they're also spreading more. So that, I'm not put the screenshot for that, but you can see these changes in number, especially for the epithelial. You see that pretty much all throughout, maybe except some, maybe germ cell cancer, which makes sense because germ cells deplete over age. So the chance of cancer goes down. But otherwise, if you look at the epithelial ovarian cancer, it increases dramatically between age groups, and it increases really robustly as a woman ages. So this made us think about, okay, what could be the reason for this, epidemiological change? Why are elderly women more prone to ovarian cancer and metastasis? So this here, I want to give a background to a theory which was proposed by Stephen Paget. It's called the seed and soil hypothesis. It's one of my favorite cancer theories of all time. When when this cancer field was really starting to develop, people were thinking about, okay, there is this healthy cell, chemical mutagenesis happens, or some sort of mutagenesis happens, and it becomes transformed. Depending on the severity of the mutations that has occurred to the cancer, it's going to determine how invasive it is, or how far it will be able to go to the tissues. So this was like a very primitive approach to understanding cancer, where people thought that it's the seed, which I'm referring to, which seed being the cancer here, where mutations are happening in the cancer, and that itself would determine how invasive the cancer would be, where it would go, and all of that. But Page had came up with this idea that not just that, what is happening to the tissues where the cancer can migrate also determines the invasiveness of the tumor, which actually makes sense. Now, when you look at all the research that has been done in cancer field, you see that so many kind of cancers have this organotropism. Ovarian cancer, as I said, primarily metastasizes to peritoneum or to the omentum. Breast cancer happens to generally localize to lungs, and so on. So this kind of suggests that there's like a soil part, which is basically the tissue where this cancer can metastasize, and what the environment looks like there, determining what the rate of metastasis or how invasive the cancer would be. And so I read this and I was like, okay, what if the reason why elderly women are more prone to cancer is actually because of aging of this microenvironment or the soil region, and has perhaps nothing or minimal to do with the mutations that are driving the cancer. And I looked up a bunch of papers and I was like, oh, there's a lot of changes happening in aging in the secondary sites. For example, the peritoneum. There's a lot of change in the extracellular matrix. With aging, the senescent cells, which I'm going to be referring to as frequently H cells, which are these cells which have accumulated damage, have become really elongated, and I'll show you some pictures of them. They kind of accumulate with age, and their number goes on increasing. So clearly, there are a lot of changes which are happening upon aging into the soil region, into the microenvironment, into the peritoneum, which has the mesothelial cells, and so could that be driving, this greater spread of ovarian cancer? So this was my hypothesis, which I started with. And so to observe this, I started with the in vitro cell culture model, where we had the mesothelial cells, the MET5A. You might see these labels. Basically these are the healthy cells which lines the peritoneum and are the first defense to cancer when the cancer tries to metastasize. So these are the healthy cells basically of the tissue. And SCOF3, which are the cancer cells, are the aggressive epithelial ovarian cancer. So we transfected them so that we can visualize them. We made the MET5A cells RFP labeled and the cancer GFP labeled. And what I did first was to just make a monolayer, because that's how the cells are on the peritoneum. I just added these RFP labeled cells, the mesothelial cells, and they made a monolayer first. And then once they have established a monolayer, I added the cancer cells on top of them and co-cultured them for 72 hours to see how cancer progresses. So this is like a mimetic of how perhaps metastasis is happening in vivo in real time in human body. So I was trying to mimic that. And one thing which I did, so these healthy MET5A cells are a transformed cell, but they're young. I can make them aged. I can make some of them aged. So I can make maybe 50% of them aged to mimic like how aged the person would be. Because with aging, senescent number increases, senescent cell number increases, which are the H cells. So I can chemically induce senescence in these untransformed cells to make them senescent. And that way I had a monolayer, which was the young monolayer, and an aged monolayer, which had, say, 50% of H cells. And then I added cancer cells on top of them. I see a question popping. How are you defining aged in vitro? That's a great question. Yes, in vitro, it's a little hard and tricky to define aged. When I say aged here, I'm looking at biochemical markers of the cells which have stopped dividing, which corresponds to senescence in vivo. So there are a lot of ways you can look at, you can do a lot of staining. There are molecular markers to P21, SA-beta-gal staining to look at if their cells are senescent or not. And we see the same in vivo and in vitro. But I do agree it's slightly different because these are not primary cells and they're immortalized, so it's hard to define real aging. And that's why we did a bunch of in vivo experiments too. But this is a very simplistic model, so I'm trying to keep it very simple. Might have missed this, but what does transformed or untransformed mean for these cells? I should have probably simplified that. Transformed here means just cancer. So SCOV3 are malignant cells and MET5A are healthy mesothelial cells, the cells where the cancer migrates to in vivo. So we've taken the primary cells and just use those. Awesome. So I was really interested in seeing how cancer proliferates within the aged monolayer and the young monolayer, and how they're different to really test my hypothesis if within the aged tissue, could cancer be proliferating faster, or could there be other changes? And then I did this experiment. So this is the data from the paper. We're going to look at the lift first, and then I'll show you a video of what is going on. What I found was that MET5A cells, which are labeled in red here, where you have the young tissue, and then cancer cells are all added on top of them at 0-hour time point. So we're taking a snapshot at 0-hour time point where you can see these spherical cancer cells. They're spherical because they're suspended, they're just going to adhere. So they're just ready to adhere. And then you see after 72 hours, they have grown a little bit and then, made some space for themselves. While in the aged tissue, you can see that the growth is way more and is more pronounced. More specifically, you also see these, clearance of the red cells, the age cells, which is shown here in the graphs. You can see on control here means the young. On the young MET5A, cancer proliferates to this extent and is kind of stabilizing at that. While on the senescent or the aged tissue, you can see the cancer growth is way higher, and it's almost constant at that rate. While if you look at the area of the monolayer itself, you can see that the area is expanding because these cells are dividing too in control, which is the young tissue, but it starts to decrease. It starts to decrease when cancer cells are present within aged tissues, which kind of suggests that maybe cancer cells are clearing or are making space for them in aged tissue to proliferate better. And I want to show a little bit of that here. Oh. Okay. So this is an aged cell, and it's surrounded by cancer cells. And you can see that cancer cells divide around it and kind of chips off the aged cell. Let's play that again. So the aged cell actually gets extruded off the substratum and then is killed. I start to see a lot of these killing to perhaps to make space so that cancer cells can grow in that space and divide further. And I asked, so these are the observations that I kind of made, which I've listed here. And from these observations, I wanted a project because that's what I wanted to work for the next 2 years. So these were some of the questions that were really interesting to me. First, I kind of saw, or I had some preliminary idea that cancer cells are getting attracted to aged cells within aged tissue and are perhaps preferentially eliminating them first. So they get attracted to the land basically around an aged cell, and then they kind of divide there and clear off the aged cell to make space for them. I mean, it could be a wise strategy to do that too, because as you can see, aged cells are way bigger. So perhaps eliminating them first is more beneficial for the cancer cells. Another question which was very interesting to me was that by that time I had started to read up on cancer cells eliminating other cells or in competition, and there are different pathways or modes by which cancer cells can do that. So I got really interested in that, and I asked if cancer cells landing somewhere and just proliferating uncontrollably can lead to pressures and can lead to popping up of cells around it, and is that how? So I would call that a passive mode, where cancer cells are clearing cells around it or far away from it by passive mode, just by dividing. Or is there an active process where cancer cell finds a cell which it wants to kill and maybe by biochemical signaling or some interaction or by shedding contact, it is able to clear off the AIDS cell. So that I'm calling that side an active process. And then the third, which is something which is really important, which I feel is one of the most important reasons why I wanted to work on this project, is it really the case that cancer cells remove remove the aged cells to create space for them, and that is necessary for the increased proliferation of cancer cells in aged tissues, because that's what we see, right? And in aged tissues, we see this increase in cancer proliferation. And that would be like a loop back to the stats model, where in the statistics, we saw that, in elderly women, there is higher proliferation, there's higher incidence of cancer. And I thought that would perhaps, explain it all. Awesome. So I'm going to go one by one to these observations and the experiments I did to actually make it into a project. So the first thing I did in my time lapse is to just check what happens, or when I look at, let's say, when I look at a young tissue and an aged tissue. I see a question. Are there empirical data about the extent of tissue clearance? Great question. I would say yes, there is empirical data about tissue clearance in cancer patients, but there is not really much. I don't think there is much record about the age related to that. People have known that when cancer cell lands on the peritoneum, especially with these cells guarding, they make these beautiful looking these cleared patterns where cancer cells are just dividing in a loop and clearing off the vicinity. So it makes these beautiful patterns. But I don't think there is much data available on how that looks, or what fraction of it is different in, let's say, aged patients. Awesome. So for my experimental setup, I basically looked at, in young tissue and in aged tissue, I looked at aged cell and young cell and counted their neighbors, and then like the neighbor cells. And then in those neighbor cells, I counted how many cancer cells are there. And I just plotted the percentage of cancer neighbors. And right from that analysis, I could see a stark change. You can see that if you just look at an aged cell, its probability of having a cancer neighbor is way higher compared to a young cell. Another interesting observation was that in the aged tissue, I told you not all the cells are aged, only 50% are aged. I don't have the graph here, but if you look at the young cells in the aged tissue, you will see much lower percentage of cancer neighbors. In fact, there would be no cancer cells around it, because all those cancer cells would have migrated to the aged cells. So we saw like this, we saw these observations, and we were sure that there is something going on. Cancer cells are getting attracted somehow to the aged cells. For the second question, I want to just give a little bit of background. So I was interested in figuring out by what mechanism do cancer cells extrude or clear aged cells? Is it a passive process where cancer cell lands, it divides, and by pressure it just extrudes everything? Or is it a more active process where cells divide, move to preferred sites, and then actively eliminate cells? And I hope you can appreciate by now, by answering the first question here, it already looks like it's an active process, because cancer cells are getting attracted to a certain cell type. So a little bit of background. The way cells generally grow in a dish or in body is through this process called contact inhibition. When I say cells, I mean normal cells, where you have cells make these junctions, tight junctions with each adherent molecule, which is a protein. Let's see another question. What in a senescent cell are you able to track with coloring, that is, between old and young cells experimentally? Awesome. That's a great question, because within a single aged tissue, I have both of those cells, and both of them are labeled red. I am not specifically counting aged cells, but by looking at, then as you will see in the next slide, where I have data for actually looking at how many deaths are happening in aged and young tissues, I'm actually just looking at total number of deaths. And if they're half-half, I'm assuming in the beginning, the percentage would stay same and then it will change. But before the cell is killed, you can look at how the size of the cell and Senescent cells are around 3 to 4 times bigger. So you can make out if it's a senescent cell or a young cell based on that. You can also do other sort of staining, which I was telling you, telling people about the SA beta gal staining to figure out which, if it's a senescent cell or if it's not a senescent cell. So this way you can track which cell is it. Is it senescent cell or a young cell? And obviously for cancer cell, you can look at the fluorescence. Just want to be, okay, we have a lot to cover. I'll move forward. So generally the cells have this uh, have this pattern where cells will grow to a density, and once the density becomes high enough that it's confluent, where cells are touching each other, they stop growing. And you can see that from this red trend. However, cancer does not either has mutations in e-cadherin or has other signalings changed that leads to non-disrupted growth, where you have an uncontrolled growth phenotype where cancer cells keeps growing at the same pace. They would grow, make it full confluent, and then grow on top of each other. And so I can implement. I started to see that in my experiments too. And one of the things that you can observe because of this uncontrolled proliferation of cancer cell is this mechanical elimination. So remember I was telling you about passive and active kind of elimination or clearance? We can see that here. I've tried to visualize it. I've tried to visualize it using some sort of density design. So I hope you can appreciate that when cancer cell is dividing, you're going to have compression of other cell types which are around it because it's dividing uncontrolled. And because of that, you can have extrusions which are happening. So a cell is getting contracted and it becomes so small that its bottom is no longer touching the substratum or is minimally touching the substratum, and a little bit of shear force could just chip it off. And so I'm calling that kind of elimination mechanical elimination. And a contact-based elimination would be that where cancer cells actually come in contact with the cell and either biochemically activate it to undergo apoptosis or actively, with its protrusions, try to chip it off. So that is more of a contact-based elimination. And these are the two methods I thought of how this killing could be happening. Two things which I want to point out, which are the major differences between these two types, is that contact-based elimination would definitely have a neighbor. You will always have a cell being extruded or killed with a neighbor as a cancer cell. However, in a mechanical elimination platform, you can have extrusions which are beyond, which are actually not in contact with cancer cells. So that was one of the things which I was looking at in my experiments. And then the second one is that if it was a contact-based elimination, and if it was attraction-based too, you can imagine that cells would not need to be confluent. The monolayer would not need to be confluent, and cells would be eliminated even under lower confluent conditions. So taking these two metrics into account, I just looked at first of all, where the cell death is happening. I looked at when the cell death happened, was it close to the cancer? Was it far away from the cancer? And I have shown that here in young tissues and aged tissues. Contact means cell death happened in contact with cancer, and far means it was distal from cancer. And you can see in young, there's minimal death. However, in aged tissue, you see this really robust increase in deaths in contact with cancer cells, which from the images, you can further appreciate that it's not a super confluent conditions. There is some amount of gap between cells. So it's like sub-confluent or reaching confluent conditions. It's not by mechanical or pressure related schemes that you're actually getting this extrusion. There was a question. Wonder a little clarification on whether senescent cells do automatically extrude themselves because of blebbing, etc. Is it specifically happening with cancer, or how can you check that? That's a great question. We actually looked at that in one of the graphs which I showed you, where the area of monolayer decreases. There was another curve which I did not discuss, which was not having cancer, but just aged tissue on its own. And it does not grow as fast as the young one, but it still grows, and which makes sense because aged cells do not divide. So you only have 50% of young cells which are contributing to the growth. But without cancer, you do not see an extrusion or killing. or if it's there, it's minimal. While with cancer, you see this really severe effect. Awesome. So the third observation, which I was really interested in, was the clearance of aged tissue, if that is actually required for the increased proliferation of cancer cells in elderly women. So before knowing the mechanism, really, you can't really reach or answer this question because, for example, one of the experiments which I can think of you would want to do with this is have the AIDS cells, put cancer cells on top of them, and then now maybe if it's through apoptosis, inhibit the apoptosis so the senescent cells or the AIDS cells do not get extruded, and now see what happens to the growth of the cancer. But at that point, we had no idea about the mechanism. So it was very hard to really devise an experiment to answer this question specifically. And this is where CompuCell came into really, really super help. So when I heard of the CompuCell platform, I was like, oh, okay, this looks interesting. And then I started to learn a little bit. And in the summer course, I learned about modeling a little bit. And I thought, what really is an important problem, or how do you define a problem which is a good problem for virtual tissue modeling? And hopefully I'll convince you all that all of these are actually excellent problem for tissue modeling. I think there was a question. Awesome. So I'm done with the background and the experimental findings. We're going to move to what defines a good problem and how do you employ those and what are the things needed. Before I move on, I'm going to just take a small break and check on questions. Okay, awesome. There are no questions. If anyone has any questions, I can take them now, or I'll move forward. Great. Silence means no. Okay, so what defines a good virtual tissue model? I've put on one of the first things which I think is needed for a virtual tissue model. So if your biological problem or your experimental problem can have these physical properties which can be measured and easily implemented, let's say in CompuCell 3D, you can ask those questions using tissue modeling. I'll give you an example in the scenario which we have discussed so far. I knew the size of AIDS cells is bigger than the other cells and cancer cells. I can measure them. So I know the relative size of these cells. That can be implemented. I know that senescent cells do not divide. I know that cancer cells divide, but divide uncontrollably. I know that young mesothelial cells divide, but show contact inhibition. That can be implemented. I know, so when I put the cells together and they grow, there is a size when they are sub-confluent. And I know once they have fully become confluent, I can measure their size and see how much are they compressible, what is the pressure, get an estimate of or a proxy for how much pressure are they experiencing. So that can be measured. And then from literature or from your own experiments, you can figure out if there is one cell secreting something and the other taking it up or getting attracted. All of these things can be physical properties which can be implemented in your model, which are known in your biological problem. Some cells getting clear or cell death can be implemented. You can figure out you can check a neighbor of a cell in CompuCell 3D easily by 3 lines of code. So all of these things can be easily implemented in a virtual tissue modeling platform. And knowing that these things are available or accessible in your biological experimental model is useful. The second thing I think that you need is really the crux, the rules. What is going to govern your simulation? For example, I talked about growth. I know that one cell doesn't divide, so that's a rule. I know that some cells divide, but they show a contact inhibition paradigm. So that's another rule. And this way, I can go about building rules for each cell type or similar for similar cell types. I can talk about if there is one cell secreting, the other taking it up. What is the rule between them? Are they going to repel each other? Are they going to attract from each other? Are they going to be, no effect stage? For the stimuli which I talked about, where clearance happens through cancer, there are 2 patterns: the mechanical clearance or the contact-based clearance, or I'm sometimes going to call it perimeter-based clearance. That's another rule that can be employed, right? So thinking of what are the things that are being shown in your experimental conditions, which can be implemented as rules or subset of rules in your tissue, in your virtual tissue model is useful. Just want to give you, and I think once you really start thinking about it, it would not take much time to develop thoughts. And with the great support, I think, especially with the great support you guys have from the summer school as well as the hackathon and beyond, I think you guys will be able to reach to good state. I just want to give a screenshot that I took from my laptop, which was when I started CompuCell 3D Summer School, maybe 3 or 4 days in, probably similar to what you guys are in right now. And this is what I ended with after hackathon. And then obviously I worked, on it to perhaps for a few months and then, publish the paper. But what started with just 2 cells sitting around there doing nothing. And then by the end of the hackathon, with a lot of incorporation, a lot of help from my friends, a lot of help from CompuCell 3D TAs, as well as people around me, I was able to get cells dividing. I was able to get cell types associated. I was able to get one cell secrete something and the other take up something. So all of these things were going on. And I feel like you guys can do that too. Okay, so we have partially covered what are the important rules and things you want to think about before reaching and deciding to do a virtual tissue model problem. I'm going to talk to you guys now about the code, the precise code, as well as what you're going to be seeing in your exercises and how the model is set up. I'll try to make it interesting. I personally don't like to look at a lot of code, just screenshot, but hopefully it'll be fun and you guys will be able to understand most of the stuff. I'm happy to take any questions right now. If anyone has any questions, concerns, doubts, clarifications, comments, feedback, anything. Okay, I don't see anything, so I'm gonna move forward. Can you guys hear me? Can someone confirm? Just want to make sure.
Yes.
Awesome, thank you. All right. So I think most of you guys are familiar. Okay, I see a question pop up. Okay, awesome. Thank you. Most of you guys are familiar at this stage, I hope, with the Hamiltonian function. Even if you're not, it's all right. There's nothing much on this slide. But we know that how CompuCell 3D works is by minimizing the energy function, which is guided by the Hamiltonian function, which has 2 parts to it. I've just kind of given you a visual representation of what those parts really mean and how they look like when you simulate. These are some of the basic stuff that I've implemented in my simulation. So by looking at this, you'll get some idea or some visual appeal to what exactly you tweak around to make a simulation. Very simple stuff. So one of the things the Hamiltonian function has is the volume constraint, which refers to where basically you have the potential energy of the cell, where if it gets compressed, it's not happy and the energy is increasing. And that factor depends on how compressible it is, which is the lambda volume factor here. So when you have volume constraint set up lower, if there is a high pressure scenario, a cell would get compressed easily if it has a lower lambda value, which is shown here on the left. And on the right, you can see if it has a high lambda volume value, you would see that the cell is incompressible almost. For the contact energy or the adhesion energy, it's basically the likeliness of how cells would like to react to each other. Would they like to share their boundary? Would they like to be in close contact or not? If the contact energy is really high, keeping them together would increase the energy of the system. So it tries to keep them away. So the cells would actually be far away. They would likely not try to share contact with each other. While a lower contact energy preferentially stabilizes cells to share contacts. So this is a visual representation of what we're going to be looking at somewhat in our CompuCell 3D simulation. Awesome. I'm just going to play a screenshot of when you're going to play the CompuCell 3D code, what all will pop up. Let's see. Let's see if it goes through fully. Okay, good. So on the left top is the cell field here. cell field is, I see it. Oh, thanks. On the left top is the cell field where you have different type of cells. And I'm going to be talking about what cell type is what. It should be pretty evident by now. I've tried to keep it color-coded, where cancer cells are in green, big red cells are the senescent cells, and the young cells are in pink. And on the right, as well as there, you actually have three graphs, which I have tried to just keep it there so that you guys have a fun interactive session in the exercises. We're going to be using these graphs to look at different models that we will try to simulate and see what happens to the different kind of, for example, what happens to the cancer cell number when you have model 1 versus model 2 versus model 3 versus model 4. We're going to be looking at also the area of cancer cells. We can look at the area and see which model leads to highest amount of cancer cell area, and what all kind of rules make that happen. And then we try to make sense of that experimentally, if that makes sense or not. And then we also have, this is only for fun, the number of senescent and cancer cell contacts and how that is changing in real time. Where if you see a lot of contacts, that tells you that there's a lot of interaction between senescent and cancer cells. Awesome. Okay, let's dive into the code. As I said, I am not a big coder, but I do have a big gallery of memes about coding, which I'll just put in here. I'll send you the slides if you guys also appreciate a lot of memes. But yeah, I'll give you guys maybe 30 seconds while I sip something to take this all in. Okay, I'm gonna move on. So this is how the thanks. This is how the code is implemented. I have 3 cell types, and I also have put in a wall here, which is a frozen wall. The wall basically, I didn't want my cells to move I didn't want to have continuous boundary conditions where cells move from left to the right. Because when you think of a system, let's say in vivo, where you have a system where cancer cells are being transmitted, they're kind of stuck in the cavity, right? And they're going to land on one of the walls. So I just tried to build a wall outside and then have all the 3 cell types between. I have the medium here, which is just normal medium. There's nothing in it, nothing getting secreted as of now. You guys are already familiar with this. I've used the senescent mesothelia terminology and non-senescent mesothelia. Senescent being the 8 cells. When I'm referring to senescent mesothelia, I mean the 8 cells that implemented as sen for senescent in the code. And non-senescent is the young cells, basically. And then cancer is cancer, and wall is the wall. I also have a small schematic here. This is how they would look in the simulation. Okay. So my XML file actually shows the contact energy between different cell types. I've kept the contact energy with all the 3 cell types really high with the wall because I didn't want the cells to just get stuck at the wall. There's motion between the cells, and I've tried to make sure that they don't get stuck to the wall. So they prefer to stay away from the wall as long as they can until the confluency hits. And then all the other cell types have so they're equally likely to share contacts with each other as of now. And then with the media, they like to be there. So they would prefer to be, without any contacts, but once it starts, or have some contacts, but once it starts to get crowded, they would share contacts. And that would be equally likely for all cell types. So there is no preference for any cell type as of now. In the Python file, which talks about all the steppables or the classes, I have 4 classes, which we're going to be talking about today. The 5th one is also there. There's almost nothing there, maybe 1 or 2 lines of code, which is for an optional exercise. I think the 2nd one. And you guys can feel free to, try that out. But I would say that when you're doing that, comment it out, please. Just make sure you get done with the comments. Okay. So we're going to be discussing about all these classes and what they do really quickly, because that's the boring part, at least for me. Okay, so the first one is the model 2D class, the model 2D steppable. This one is pretty much the mother steppable. I started with this steppable. I was learning then, so I basically put everything in this steppable, which was stupid now when I think of it. But it is how it is. I have the layout shown here. This is the number of cancer cells, which talks about how many cancer cells are going to be in the beginning, and then they would proliferate from there. And then this is the method for the layout where I have interspaced senescent cells, and then I have young cells sprinkled or cancer cells sprinkled between them. Okay, so this region basically determines the layout. We're not going to be touching it much today. I think there is one of the optional exercises where you can try out different layout methods, but for the interest of time, we might not go there. The next one, the next code in the same class is the part where I'm initializing the sizes of these cells and how compressible they are. When I looked at my experiments, I took some idea of how big these cells are by measuring them under the microscope, and then I implemented the relative sizes based on that. I did not see, once they become confluent, I did not see any changes in compressibility, or they were equally compressible. So I just implemented some number. You would see this a lot, ranging from random integer between 40 to 58. That's meaned around some value, which is 49. That's just to create a little bit of biological noise, where obviously not every cell is going to be equally sized. So I've tried to, you'll see that a lot throughout the simulation. So whenever you see, if you're very interested in the exact size, just mean it out, just average it out. I have also implemented a low probability spontaneous death of cells, which is a very small amount. So you can see that it's a precise number 5 out of 30,000. So it's a very small number, very small probability where a random cell, any cell, can just randomly die, just get removed from the simulation, which mimics sometimes cells would just on their own die. So I just wanted to, again, to produce some biological noise and have some, real-time observation just come into the simulation. The next thing, which is interesting and is a little bit mathematical, one of my friends who came up with that, which I think is just too complex. It could have been easier, but it is what it is where I'm actually implementing the growth of cells. So I told you senescent cells do not grow, they're arrested, and they're really big. The young cells, the young mesothelial cells and the cancer cells grow, and they grow with this equation where I calculate their pressure. Pressure is calculated by this formula right here, which is a very simple formula. You're just checking how much is it deviating from its target volume, which gives you a proxy for how compressible and how compressible they are, and gives you proxy for what is the pressure the cell is experiencing. Then I'm again using a mean around 1, and I have this factor 50 by 1,000 and 75 by 1,000, 75 by 1,000 for cancer cells, because cancer cells grow they grow faster. If you just look at the division time of a cancer cell, it's in 52, so 2 to 3 ratio. And then I have this factor 1 minus something. I'm just going to call it x right now. But the value of x is actually a Hill equation, which determines contact inhibition. So the value of 1 reaches really high when you have, this is from a ligand curve. Hill equation is used a lot in ligand kinetics, binding of ligand to the receptor. So I've just taken that and I've implemented that. The most important thing here is the K. The K here is 75 and it's 1500 for cancer, which shows that cancer cells will not show contact inhibition, or will be very little affected by contact inhibition, even to such high pressures. While these guys, the young ones, will start to show contact inhibition at a very low pressure. We can talk more about it if someone is interested. And then the most important thing, which is the contact-based clearance rule, which I have implemented as shown here, where its default state is false. We'll make changes to this. The threshold, which means that how much percentage of the perimeter needs to be covered by cancer cells for cancer cells to clear an AIDS cell, is shown here. So 50% or more would just kill the cell. If it has 50% of its area covered by perimeter covered by a cancer cell, you would just set the target volume to 0 and for a senescent cell and make, this is one way of killing the cell. You can just say cell.death. There were some pixelated issues, so I just implemented this. But here we are calculating the area that cancer cells share for each cell type. And once I have that number, I'm dividing that to the total cell surface or the cell perimeter, and then checking if that value is bigger than 0.5. And if it is, and if the clearance rule is true, is implemented, we will kill the cell. So this is the most important thing. The other thing, which I'm not going to go into much detail, is the random motion of cells. We can talk more about this, but we're not really going to play around with this much. So I'm just going to skip it for now. But it basically means that I'm randomly finding a theta, which basically is the angle at which the cells will move. And then these cells move in a direction based on that. And that is what is implemented in these two pieces of code. The third one is the growth and mitosis, pardon me, growth and mitosis class. And it's a misnomer because growth has already happened in the first class. We're only dividing the cells here. And again, very simple. I've just doubled the volume. If the volume is double, that's the cell target volume, then you should just divide the cell. And that is what is implemented here. The third one is the plotting class, where I'm plotting 3 graphs. First one is here, where I'm plotting the number of cancer and senescent contacts. This is just implementation. The exact parameter calling and what values are given for the graph is in the next slide. And this one is the area implementation, and this one is the number of cells implementation. This is where parameters are being referred to the plot. We are calculating the average cell neighbors, where average cell neighbors is actually being shared across different steppables. So it's being calculated, I think, in one of the steppable, probably the biggest, the steppable model 2D. And I'm just calling that and referring that to the graph to plot it. Then for the cancer cell area, I'm also calculating that somewhere, probably in model 2D, and just referring that. Same with the non-senescent area, senescent area, and so on. For the number of cells, I'm just calculating the length of the list, which entails all the senescent cells, and so on with non-senescent and cancer. And the secretion and uptake class is right here, which we might or might not use. I think we're running a little short. I think we're good. We might use it. We'll see. But it basically just initializes the secretion field called chemokine, which is a fancy term for a cell secreting some molecules. And you can, in one of the additional exercises or extra exercises, you can write your own code to actually make one cell secrete. I've given some instructions in the slide as we move forward. Okay, so that was the, yes, yes and no. So in the experimental setting, what we found is that SAS, in this scenario, you can imagine that it is SAS, but it's the senescence-associated secretory phenotype, the interleukins or chemokines, specifically maybe IL-6 or something. But as you would see, that's an extra exercise because that was not part of the model that we submitted, because we later figured out from experiments that SASP is not what is leading to attraction. It could be the extracellular matrix, so it's something different. Any other questions before we move on to exercise? Okay. Okay, so we're ready with the exercise. I think before I really start, I just wanna, okay, before I really start, let me just, how about I pull out this, I pull out the code on the tweeted and I try it myself so that. Okay, before I start, let me answer a question. Looking back at this from 3 years on, what are some key assumptions you made and how did you justify them? Oh, I would say I played around a lot. There were some things which I played around and I was like, oh, this looks really cool, but it had no sense. So as in like, it did not make any sense. I just went, I mean, I'm showing you guys the model which was published after 4 to 5 months of, tweaking around, playing around, getting feedback from people, getting feedback from my PI, getting feedback from other people. So there were a lot of assumptions that I made and some of the things which I just tried. And I think I don't explicitly remember what assumptions were discarded because they're no longer in my brain anymore. But there were some assumptions that did not make sense and they were being discarded. So when you're actually going to have a model ready to, submit to any platform, you want to make sure that things you're implementing are there for a reason. Awesome. So what I would do is I would just open the file and I would try playing the simulation once, and then I would make you guys play the default simulation and we'll move on to exercise one, which entails default simulation and another parameter. Awesome. Can you guys see my screen for, let me actually close this. Let me start from scratch. Okay, so we're going to go to I hope you guys have received the zip file. You guys can download the folder. They should be labeled very similarly, so it should not be confusing. You should start with the BVT OFCA aging model, the first one. This is the default one. We're not going to be, or we might, if we have time, use these two. So these are the three zip files. You will use the first one to get the first model and open that. So these are the three files that we have. And I'm going to play that in player. Okay, when you play that, you will see a lot of windows. You might have to rearrange that, hopefully only for the first time. Okay, while that is running, let's move on to the first exercise. I want you guys to download the first folder which I talked about, and play the first model, which is the default model, without doing any changes to it. And then the next thing I want you guys to do is to go to the Python file, the main Python file, which has all the description, and change the clearance rule from false to true, which is shown here, and then play the simulation again. And before you go ahead and play the perimeter clearance model simulation, we are going to actually collect data because that's how the life of a modeler is. We're going to collect some data, and I want you guys to really experience that, when you build models, and obviously we're only going to do that once. I have optimized the simulation. It only takes max 2 minutes. So I've changed the parameter so that the simulation runs really fast and the cells grow really fast. So you guys should be able to run a lot of simulations in the time we have. So play around with it, play the first play the default model and make sure that it's working. Then once it's played, so it's done already, as you can see. I want you guys to go to each of these graphs and eyeball the measurements. So you can zoom in and get an idea of what value it is. And for the data that you guys are going to collect, this is the part which shows. First, we are going to you feel free to write it on an Excel sheet, or if you have a paper, write down the name of the model that you used. Write down what was the cancer cell number at the end of the simulation, and what was the cancer cell area. Then look at how many senescent cells were remaining at the end of the simulation. If the simulation, I don't think you guys, this might or might not happen. But if the simulation ends with 0 senescent cell, then figure out when was it that at what MCS did the count become 0 for senescent cell. And then also calculate the maximum value of senescent cell, cancer cell contacts, which is kind of being tracked in real time throughout the simulation. So let's do it with me. So this is the simulation. It is played already. One of the things I also want you guys to try before you record data is focus on the simulation. If you just play it's only 90 seconds. Just look through it, look at if there are any patterns, look at if cancer cells are moving around, if there is maybe cancer cell, touching another senescent cells, all the cancer cells are aligning with the senescent cells. Are they killing the senescent cells? Are senescent cells getting too compressed and being killed? All of these things, like basically make any observations if you can. And once the simulation is done, record these values. So for example, for the area, for the cell number, red ones are the senescent cells. I'm going to zoom in here. 18 is the count at the end of the simulation. So record that. So senescent cell number is 18. For the cancer, you can go here. You can also just eyeball it. If you see it's like maybe 122, just write 122. And for the cancer cell area, you can write maybe 12250, something like that. But basically eyeball it and make a note of it. Is there a question? Sorry, I should keep I should keep this. I'll keep this slide on while you guys are running simulation. Let me know if you need any help with the simulation, or if something is not playing. I'll let you guys work on it for maybe, since we are simulating two models, default model, and then I want you guys to make this one change, which is change the clearance rule from false to true and run the simulation again and record the same values again. So I'll give you 5 minutes to do it, and then we can talk about the results. We can talk about if there were any observations or anything you guys want to discuss. Yeah, feel free to just shout out for help anytime you need. I'm here. We can also check if there's something not running or if there's something um, wrong. And while that is in place, if people find that is very easy, this is the optional exercise, which is a little harder. You guys will write your own code here. It's the extra exercise. I've just written it here. I'm not gonna distract people who are doing the simulation for the first exercise by screaming all the things that you're supposed to do here. It's very self-explanatory. If you just go through it, you should be able to implement this. I might or might not help you guys in this based on the time that is there, but I've also shared the solution with you in the zip file which says extra exercise one optional, something like that. The same code with the new mechanical clearance rule. And I will discuss why this was important once most of the people have finished recording these values for exercise one. Let me know if someone wants me to go to the optional one. I can change the slide. And obviously, when you're running simulations for real-time experiments where you're gonna, measure values, you're gonna do it in like 10 tries or 15 tries and take a mean and all of those things. We don't have time for that, so we're not gonna do that. But yeah, hopefully there will be less variation, and we should get something out of this based on the data you guys collect. Are you guys having any difficulties? Is there anyone who has completed getting the values? Oh, I see them there. Okay, thank you. Thanks for putting them in chat, Hayden. Okay, I'm gonna move forward. I see some of the responses in the chat. Thank you for sharing that. Yeah, the ideal way would be to actually, we will not do that, but actually just because there are so many people trying, or there are some people trying, we can get some values and take a mean or something, but we're not gonna try that. That'll be too much time. For the optional exercise, I'm just gonna discuss this and how one would implement something like this. Remember I talked to you guys about, this is the contact-based rule that you guys just tried, where clearance is happening by calculating the perimeter of a senescent cell. So you look at the perimeter of the senescent cell, figure out how much of that is shared with the cancer cell. If the sharing of the perimeter crosses a threshold, you kill that cell. Now, this is like you need a cancer cell around a senescent cell to kill it. The other clearance rule, which we discussed in the background, was the mechanical clearance rule, where it doesn't matter if a cancer cell is around a senescent cell or not. Because of the sheer proliferation pressure of cancer cell, you're compressing other cells, and you're able to eliminate cells based on that. So the way I would implement it first would be to make a cell more compressible. So I'll make the senescent cells more compressible. Remember the lambda we talked about, which was distributed equally? I think the mean was around 20, something like that. I would make that maybe 2 or 4. So that senescent cells are more compressible. The second thing I would do would be to remove the perimeter rule. So you can either comment it out, delete it. And then I would check, so I would go to a for loop across all the senescent cells. I would check what is the exact volume of a senescent cell at that MCS, at that Monte Carlo step, and take a ratio of that with the total volume of the cell that it's supposed to be, so the target volume. And that's your fraction now. If that fraction goes below a threshold, let's say 0.2 or 0.5, as it was in the perimeter rule, then you're killing the cell. So that's a very crude way, a very crude and simple way of implementing a compression rule. One thing I would do, if you actually go ahead and do it, would be to increase the MCS slightly more. Right now, the max MCS, the simulation stops at, I think, 5,000. You can make it 6,000 or 7,000, so that, as you can imagine, mechanical rule will start to play in a lot when there is super confluent conditions. So you can let the cancer cells grow a little more. And then you would see that all the cells, that's my understanding. You would see that. I think I've tried it when I was tweaking around with the parameters before, and you will see all the senescent cells being cleared, but you won't see cancer cells in vicinity clearing them. You would see them getting cleared even for the senescent cell, which is isolated away from cancer. And that is not what we see in experiments. And so that's why that model is not, or that clearance rule is not applicable here. Awesome. I'm just going to share the results that when I had played it once, I got. So this is a This is the default model. This is a fast-forwarded screenshot. These are the values that I got. I just put it on an Excel sheet. I've called it no rule model because the parameter-based rule was off in that. You see cancer cells are growing, but you see that most of the senescent cells are still there. In fact, I think you start with 25 and all of them are still there. Cancer cell number is 101. This is less useful, maximum of cancer cell and senescent cell contacts. For the other rule, when you have perimeter-based clearance, you start to see, as you will see here, maybe this cell. Yep, that cell popped out. So let's see. This cell and this cell popped out because they were sharing a lot of area with the cancer cell. But that only happens when there is confluence. It has already become confluent here. So this is already confluent conditions. In experiments, we have seen that it happens in sub-confluent conditions. And you can see that the senescent count is lower here, 14. So I guess 11 of them did not make it. They got eliminated by cancer cells. Cancer cell area is higher, cell number is slightly higher, which suggests that cancer cells were growing more and they cleared off some senescent cells only in confluent conditions. So that's important because that's not what we see in the experiments. A question. By mistake, I ran the extra exercise simulation first. Zero contact. Oh, I see. But when it is completed, I'm seeing a 0. Yes, so the contact side between, so you're supposed to take the maximum of that curve for the contact between senescent and cancer, not at the end. It's the maximum throughout the curve. And it's okay if you have used the extra. You can try don't use both I would not open both of those simulations together, like on TweetIt. I would close one project and open the other. Oh, it's 0 throughout. Yeah, I'm not sure why. If we have time at the end, we can discuss it. But for now, I'm gonna move forward. Let me know if it's all good. Let me know if let me know if none of your models that you're playing have any contact. If it stays 0 for all the models, let me know then. Thank you for pointing that out. Okay, so we're gonna move to the second exercise. Here, one of the first things that I would do would be to turn off the parameter rule again. We are building 4 models. We have made 2 of them. 2 of them are remaining. The other one is the attraction model. But when you're implementing attraction model, make sure you turn off the parameter-based clearance model. So we'll make that true back to false again in the clearance model. And then we will go to the XML file and change the contact energy from 30 to 15 for senescent and cancer. Make sure you do that for senescent and cancer. So you're lowering the contact energy between cancer and age cells so that they can share more area, more parameter with each other, which mimics kind of senescent cells having more receptors for cancer cells or something like that, or like a basically adhesion sort of model, which I've called the attraction model here. I guess it should be called, to be very precise, it should be called adhesion model maybe. And we will run those simulations. So we'll try it with just the attraction model first. And then again, we'll go to the parameter-based clearance model and turn it on with attraction model on. So this way we have 4 models, 2 rules, all permutations done, off, off for the first one. The second one is the parameter-based model, which you turned on, which is already done. And now you'll do the attraction model individually and then attraction plus parameter-based model. Actually, let me go and run that too for you guys while you guys do it. Make sure to hit save before playing it, because if it says red here, then it's not saved and you don't want to play it before you save it, because then it's the same previous model. Okay, I'm gonna rerun this and we're gonna see what happens. One of the observations which I can make right now already is that you can see cancer cells are really loving it with the senescent cells. They're making these rosette-like structure or intercalation kind of structure where a senescent cell is surrounded by cancer cells throughout. And as a consequence of this, you can see that contact sites between senescent and cancer is already popping. It has crossed 100 easily. And it continues to pop, which is happening because cancer cells are dividing and more of them are there and more of them are sharing contact. Okay, the simulation is done. And again, I'll do the same thing, calculate the maximum. So I'll go here. I will not take this reading. I'll take this reading, which is 166. And for the area, I'll do the same thing. You only have to calculate cancer cell area. Maybe 10770. And for the cancer cell number, I'll take this reading. 117. And senescent number below. 24. And I'll write those down or put them in an Excel sheet so that we can compare between models. And now I would go to my model again and turn on the clearance model, the clearance rule, sorry. Save it. And play. I think one of the things you can already see is that before confluence is reached, most of the senescent cells are already dead. Are you guys able to run the two simulations, the two models? Can someone say or point out in the chat? Okay, if there is any difficulty, let me know. Happy to help. Awesome. I'm gonna give 1 more minute. There is a third exercise, but I think in the interest of time, we might not go to the third exercise. I would highly recommend you guys trying the optional 2 exercises. They're super fun. The mechanical rule one is really fun. The second one I'm just going to discuss. Yeah, someone has posted the results. Yeah, so the senescent cell and cancer cell contacts going low in the last model that you guys tried, the 4th model, is because the senescent cells are just getting cleared. Like they're getting killed, so you don't have enough. But in the third model, where there was a traction rule but no clearance, you saw such high values because the contacts were made, but the senescent cells were never killed. So the contacts kept on increasing because the traction is there. And that is the reason why I wanted you guys to look at the contacts. And that's how I calculated basically the contact and the other parameters which were being plotted and employed the clearance rule. Okay, so I'm gonna move forward. This is the optional exercise 2, which I felt was very fun too. Um, this is related to the secretion and uptake stepable. Um, as I said, this is more of an adhesion molecule than really a traction molecule model. Um, if you want to like employ a real attraction model, you would use chemotaxis. I have already in my code, it already has chemotaxis model, but it does not have a cell secreting and a cell taking up the chemokine. You can make that happen. You can just go to, so if I go to tweet it, you can go to the CC3D Python and go to the chemotaxis, not chemotaxis, secretion and uptake. And you can make a cell secrete outside the cell at boundary, let's say. You click on that, you get a code, you employ that in the secretion and uptake and make those cells senescent, and then make cancer cells take it up, maybe uptake inside cell at boundary. You tweak around with the parameters a little bit, and you basically go decomment this, which is the secretion and uptake stepable, and then you go to the chemotaxis plugin, which is already there in XML, and use a field use a force for which corresponds to your lambda between senescent and cancer. Try 50, 70, try different values and see what happens. And I've tried it, and it's pretty fun. You would see that a cancer cell actually chases the senescent cell, but that's not enough to clear them. Then you basically add more rules and see which one mimics the experiment will set up the most. This is when I had tried the Model 3 and Model 4, which you guys tried, the values I had gotten. The values could be very different, but one thing that I do want you guys to appreciate is that you would see the highest amount of cancer growth generally in this model. And most importantly, you will see most of the senescent cells cleared, which is what we think is happening when that mesothelial cell area goes down in aged tissue. And this is the most comprehensive model which describes that well. And that is happening at a sub-confluent condition, as you can see in this. Before confluency is reached, most of the senescent cells are already cleared off. One thing which you would see in the optional exercise is that sometimes you would have these isolated senescent cells which do not get a cancer neighbor. They just got isolated or shielded by the young ones. But in chemotaxis, when you try chemotaxis and attraction and perimeter clearance, you will never see this because cells are always secreting. And before the cancer really starts proliferating, it will end up migrating to these aged cells. And so that is a more advanced model where you can actually have all the rules implemented and have an even stronger phenotype. But this was sufficient to make the main claims about the observations, and we went ahead with this. This is a summary slide. This is what we started with on the experiment side. We saw cancer cells, making these contacts with senescent cells, kind of killing them. So we thought it's the perimeter-based or contact-based model of clearance of cancer. We implemented different models. I've taken the snapshots here and tried to make it easy for you guys. Model 1, where there was no active extrusion or perimeter rule, there was no adhesion rule, and this is what we get at the end. Senescent cells are there, completely fine. Don't see much clearance. Second model, we tried the active extrusion, we tried the perimeter-based rule. We see some senescent cells being cleared, but only after the confluency is reached. The third one, you see these beautiful structures where senescent cells are always, fully pretty much engulfed by cancer cells, but are not cleared off. They're just attracted. Where you have adhesion, but no clearance. And only in the fourth model, you see most of the senescent cells are being cleared before confluency is reached, which is what we think is happening in aged tissues. So These models made me conclude that it's the attraction of cancer cells to AIDS cells as well as their mode of clearance being a contact-based mechanism um, that that is useful for cancer cells to proliferate and is kind of necessary for cancer cells to proliferate in the aged tissue. There are three more. There's two more exercises. One optional, one. The third exercise is just tweaking around with parameters. That's what the life of a modeler is. For the contact energy, I made you guys make it 15. You can make it even lower, or from 30, you can make it 40 and try out things. For the threshold, where what percentage of the parameter does the cancer cell need to have to clear it, you can make it really low so that easily cancer cells can chip them off, or make it really stringent and try out a few things. For this one, I would only want you guys to look at the cancer cell number and the senescent cell number. I have I had an optional exercise too. You can play around with the layout. Just wanted to tell that layout is very important. It determines. I try to have a biased layout because when I tried random layout where any cell can be anywhere, um, you would have these circumstances where a senescent cell would get isolated, and that would lead to a lot of variation. So pick your layout wisely. You can also have a different amount of cancer titer by changing the number of cancer cells in C. And one of the things you would observe with that is as you increase cancer, clearance happens much earlier and so on. Very intuitive stuff. This was the results I had for changing the contact energy. When contact energy is higher, the number of cancer cells is lower because they're not making enough space for them. And when contact energy is higher, senescent cells number are really high because they can't be cleared. They're getting repelled from cancer and so on. So basically you play around with parameters. You figure out what mimics your experimental conditions best or what describes them and what are the rules that are important for that. And that can give you a hint on your biological problem in modeling. That is it from my part. I've shared my email here. I'll also share it in the chat. We are on time, which is surprising. Yeah, let me know if you guys have any questions, any clarifications, any feedback.
Yeah, we have a little time available for questions here. If anybody wants to unmute and ask or discuss further, you can also put questions in the chat.
I think there's a question. Is it possible to move the wall during simulation? I would like to include an external mechanical stimulus to the cell. Any idea? Oh, I see. That's a great question. Yes, wall can be moved, and if the whole idea is to actually, you can keep the wall or remove it whatever way you like it. I wanted a closed system, and that's why I had the wall there and I had it frozen. But you can definitely have a simulation where you have, in your wall you have cells on one side, and you have like a piston on one side, and you can with MCS change the piston to compress the cells to have a mechanical stimuli. I think a lot of people do that where you can take maybe on the left side or on the right side, you can just take a piston and that's perhaps part of the wall or not part of the wall or something else. It's something which cannot be compressed while the cells can be compressed, and you make it move, say, X position 100 to position 80 so that it's moving to the left. And that way you will compress your cells. And then after a point, you can remove them. This was great. Thank you so much. Would setting boundaries to no flux be equivalent to having a wall? Oh, I mean, the way I have implemented it, no, because I have actually implemented higher contact energy between wall and the cells. But yes, you can do that. The only issue with that is that because the cells prefer the medium and not each other, they would just want to go to the edge. Even if there is no repulsion from the edge, they would just go to the edge and sit there with the medium surrounding it. So they will not actually interact with each other. But that can be implemented for sure. Yeah, it's mostly I guess it's a little biased because that's not what happens in vivo. But yeah, these are some of the things that you have to think while building a model, I guess, that what are the things which you know for sure are limitations of your model? And And are they okay? Like, is it okay to have that as a limitation going forward? Uh, Repulsion. Could mechanical stimulus be implemented using step function to increase the contact energy? Ah, I see. That's a good question. I guess yes. I do know that I do remember uh, someone teaching about the steering panel or something where you can implement it in Python code, but you can play around with the contact energy between the wall and the cell types with the steering panel as the simulation is in play. And
Yeah,
that could be perhaps used.
we
I don't
should
know if
have some brief instruction on steering panel next week,
awesome.
which is helpful when you're trying to figure out if and what you can do during a simulation, because you can modify parameters as it goes, but steering panel allows you to actually basically introduce events
Ah, I
into your
see.
simulation so you can make changes as things progress. So you don't have to keep editing your code and
Right.
and rerunning and everything.
Great. Yeah, I have you guys have my email. If you guys have any questions or some piece of code you would want, feel free to shoot me an email. I'm happy to help in any ways. Thank you.
Yeah. Thank you,
Um,
Bharat. That was a great talk.
uh,
Hi, could I
yes,
interrupt for one second?
Yeah, very
So,
quickly, Subhani, let me
yeah,
just say
yeah,
real
yeah.
quick. For anybody who doesn't have any other questions, we're going to go ahead and take our 15-minute break now, and then you'll meet us back, excuse me, back in the main room at 2:45 for our final two hackathon talks for today. So thank you everybody for coming. So sorry, Subhani, for interrupting.
Oh, it's all right. Hi, Bharat. Thanks for the amazing talk. Like you explained everything so beautifully. I just
thank you.
wanted to mention that I'll be joining my MTech project under Dr. Amre and Dr. Mohit
Oh,
this
that's
year.
great.
Yeah, I'm looking forward to learning from you as well.
Sure, I am in touch with some of the people in Ramray's lab as well. I think Prasanna, you might or might not know, is also doing a lot of computational 3D simulation. He
Yeah,
might
yeah,
be an
yeah.
expert now, but yeah, happy to just shoot an email, happy to collaborate, happy to talk about anything.
Great,
I'm
great. Thank you so much.
I'm excited for you.
Thanks a lot.
Awesome. Awesome. No more questions. I'm gonna stop sharing. Have a good day, guys.
All right, sounds good. Thank you again.
Thanks, Hayden, for hosting me.